Now, it's your turn. It's wake-up time. I have a question for you. Now, you cannot do multiple selection questions with these things, but what I would like to know is, what is the main problem in your operation? What kind of pest is giving you the most issues? And if you're not a grower, please let me know what your clients say, what the growers that you visit are having issues with. And this is important. It's going to help me set up priorities for this year. So please go ahead. You can choose the choice. And you know what? I might have made a mistake, because I don't know if you actually have number 10. So if you have other issue, you don't get to input. If there is? Yeah. Good. OK. Because I kept thinking about pests and just writing them there without realizing that I might be running out of options. So please go ahead. I got 15 responses.
You want just one response for the most important?
Yes. It doesn't allow for multiple selection. But now that that is mentioned, you got a feedback form when you came in here. It's a little survey. You can rate the issues over there. You can tell me what's the most important. And if you have more than one problem, you can actually write it there. You can write any ideas for any projects that you might want for the center to do. So please go ahead and take advantage of that. I actually read those forms. I had a suggestion last year of doing a trial to test the effect of pot color and ground cover color on plant growth. And I thought it was very interesting. We didn't get around doing it, but I have it on the list. So I do read those things. So let's see what it is. And 46% had issues with thrips, and then 38% with mites. And then we go, white fly was not that big. Number 7 was mealybugs. So there's an increase in mealybug issues, and we are doing some work on that. So we are ahead of the curve. That's good. Now, the next thing that I would like to know is, what's important to you? So the first option is, do you want us to do studies on conventional pesticides only, or softer chemistry and biorationals, or biological control, product rotations of only conventional pesticides, rotations of biorational pesticides only, or you just want rotations of both types, conventional and biorationals? There's a lot of interest on natural products, and sometimes they don't work that well. Knowing if you have interest on that kind of product will help me when designing projects. So please go ahead and input your answer. Well, we had 25 last time. There we go. So it looks like we have 25. Working clickers, 27. OK, everything, huh? Well, that's encouraging. So I have a lot of options to choose from. Now, continuing with research areas. Disease control. Now, I can't say that I like disease, but this is just to show you that you can do cool things with microorganisms. They not just cause disease. And this is, I can't remember her name. Her name is Margaret. What I can't remember is her last name. She works with human diseases, and she does this every year where she puts out a bunch of pictures of Christmas themes that she makes with microorganisms. So there were 2 trials that we did. We worked with the efficacy of Trinity to prevent rhizoctonia. We did some work with Empress and Pageant for disease control, but also for plant health. And today what I want to talk is about what we did with Trinity. So the trial, very briefly, we got hydrangea liners. We transplanted them into bigger containers. We let them grow. We did applications of Trinity at 6, 8, 12, and 24 ounces per 100 gallons. We did 3 applications at intervals of 14 days. And we did 1 inoculation with rhizoctonia, and we did that 3 days after the first treatment application. And then we checked for disease severity and for phytotoxicity all the way to up to 8 weeks after inoculation. So what did we find? Our control was Medallion, and we found that Trinity was as effective as Medallion, but only up to 3 weeks after inoculation. But we did find that 4 weeks after inoculation, all plants had the disease. Different degrees of severity, but that was basically how long it took for the plants to develop the disease. We also included actinovate. It did not prevent the development of the disease, but when I looked into what could be happening, what we found is that it's very sensitive to chlorine. And if you use Actinovate, it's good that you take this into account because we were using city water. It's just potable water. And apparently, even the levels of chlorine in potable water would affect the microorganisms. And they recommended, I mean, the people that work with it, recommended to use tablets to dechlorinate water. So this is something that maybe in the future we'll test again. We didn't find any phytotoxicity, but expressed as necrosis or chlorosis, we didn't see the plants burn. We did find the stunting, though. If you applied more than 6 ounces per 100 gallons, the plants were stunted. There was a little bit of a disease effect in that case, too, because plants that were inoculated were also smaller than the non-inoculated control. But we did find that rates of higher than 8 ounces had an effect on quality, and the plants had lower quality. So this is so that you have an idea of what we saw. I mean, isn't it amazing? It has more than half of the stem covered with a canker, but it's still alive, hanging in there. This is a smaller canker. So what I want to show you is that it was difficult to rate this because the plant is alive, but the disease severity is very high. So we had fun with the methodology. But the reason why I'm showing this is because when you see this picture, you might not believe that any of those plants had it, but they did have the disease. So this is my control, inoculated control, and then the different rates. And you can see that there was an effect on the plant. They were stunted. And they didn't grow out of this at all. This was Actinovate, where it didn't have any effect on plant size, and then Medallion, which actually worked really well. And so keep in mind that it did work. In this case, it provided the same protection as Medallion, Trinity. But you have to be mindful because it does cause the stunting of a plant. More than 6 ounces was really, really bad. So your turn. What kind of diseases are a problem to you? If you could please choose the one that causes you the most trouble in your operation and just input your response. So I had 27 last time. We are at 24. All right, we'll just close it at that. So the same for crown and root diseases and for downy mildew. To be honest, I like working with downy better than working with root disease. So we might see some trials at the end of this year studying downy mildew. Now, what about the kind of plants? Do you have any particular interest? What is more important to study? Beddings and annuals, herbs, woody perennials? I got the Australians because they're becoming more popular there as well. Cactus and succulents, other, or just everything? Let's see if you're going to say everything. So we'll close it at that. So bedding plants and annuals and just everything. Well, I kind of like that. At least I have a choice, right, on what to do. Can always choose and then come back and say, you said everything. What about the kind of trials? Similar to what we just did with pests. So do you want traditional chemical methods only or biorational pesticides and softer chemistry, rotations and tank mixes of conventionals only, same thing with biorationals, or just combining everything? Okay. The reason why I include that is because I would think, for example, if your production is organic, then you really don't want to mix anything. But at least I particularly believe that there's not going to be a magic bullet, and the softer chemistry may work, but you're going to have to give them a little bit of a push maybe with conventional pesticides, unless you have a biological control program, and then you may be running into issues like what this operation did with ligas, and then find other alternatives. Now, plant growth regulators, because you don't want your plant to grow like this. And every time that I prepare this presentation, because I always include PGRs, I try to find good pictures for Pgr humor. And if you ever find one, please send it to me, because I keep coming back to this thing. It's called the corpse flower, and apparently it smells like a rotting corpse. But it's huge, and it illustrates what I want to say. So we did a little bit of work on plant growth regulators, 2 trials, one studying ojo for branching of succulents, and another one studying the effects of PGRs on branching of woody ornamentals. And this is what I'm going to be talking about today. But just so you know, branching of woody ornamentals was tough. And there was a lot of work and a disappointment because I didn't find anything that would make camellia, magnolia, and other things branch. Basically, I have to repeat it and maybe increase rates. But ojo did work okay for some things. This was an interesting trial. We chose euphorbia, different species, and these were Milai, Trigona, and Amac, because some of them are very, very tough to branch. It's still a huge interest of growers to save on labor. It's so much easier if you can just go ahead and spray a chemical rather than having people pinch. In the case of succulents, sometimes you lose the aesthetic value if you're pinching. They just look really bad. So what we did, we used sprays or drenches of the product. These were the concentrations in parts per million. And then we counted branches and height of the plants at, up all the way to 8 weeks after treatment. And also phytotoxicity to 28 days after treatment. And I'm going to show you only the results for branching. Now, Emili, so Euphorbia emili, crown of thorns, right here is the green bars. We didn't find any effect. It didn't do anything. We found the same thing with Euphorbia amac, which is this purple line. But see the increase in Trigona from 10, it went all the way to more than 25. So it does work on some things. Now they are all the same genus, so it was interesting to see how it works on some, but not others. How did the plant look? You can see here that it had an effect on height. We found the same thing for Euphorbia amac. It did reduce height. In this case, it might have been just a proliferation of branches that also had an effect on that plant. But you can see how they look. We did find that it was phytotoxic to one of the species. This was Euphorbia amac. So you can see only the drenches. And I'm going to go back because I should have emphasized that. So there was an effect with spray at the highest concentrations. If they have the same letter, they are the same as the control. But look at the drenches. They were the ones that had the highest effect. So succulents have a very tough cuticle. It's a very thick cuticle. So it's telling you that maybe the problem is that the product is not getting inside the plant. The drenches work much better. But with drenches, you have to be careful because of phytotoxicity. So what we found is that at least Euphorbia amac didn't branch when we drenched it, but they didn't like the drenches either. So this was the lowest concentration, medium, and high. And you can see how it completely killed the upper portion of the plant. So bottom line, if you want to use it, you're going to have to test it. Even if you test in similar plants, they may vary. Even plants in the same genus are not going to respond the same. And you have to be careful because it can be phytotoxic. All right, your turn. What about Pgr trials? Now, I like Pgrs, and I'm already thinking about trials to do this week. So this is particularly important for me. I would like to have some guidance on what's important to you. So do you want more trials for high control or branching? Both, height and branching, or you just don't want any more Pgr trials? And if you, by any chance, want more work on vegetables, that's some magic, please go ahead and write it in the form, because I actually like some magic. All right, we'll close it at 22. So you want both height and branching. All right, that's good, because I get a choice. What about plants? So there's a lot that has been already done in color. You know, Joyce Latimer in Virginia Tech has worked a lot on these kind of plant annuals and liners. But here, in this group, do you have any interest on more work on herbaceous or just perennials? Maybe vegetables. The only option here would be some magic. That's about it. Cactus and succulents or Australian plants. Perennials, okay. Yeah, I think that's where the work needs to be done. It's just sometimes a little bit frustrating because they are slow growers. It takes a long time. It took 12 weeks, 16 weeks for that last trial that I did, and 5 species were tested, and we had 3 products, a spray and drench, and 3 concentrations of each, and we didn't find anything. So, but we'll keep trying until we come up with something.